Welcome to the MicroGenDX Mic'd Up podcast. Discover how cutting-edge next-generation sequencing technology is revolutionizing medical diagnosis, empowering healthcare professionals with rapid and accurate identification of microbes. Our experts take you through the science behind microbiome testing so that you can make better decisions when it comes to patient care. Plus, hear stories from patients firsthand about their journey toward better health. Let's get started.
You have probably heard that in the human body, there are as many bacteria as there are human cells. Yet we have sites within our body that should be sterile. When we have an infection, it is caused by microbes, bacteria, fungus, viruses, that move into a sterile site. Or the community of microbes currently living in that body site enter dysbiosis, an imbalance between the host's microbiome. An infection occurs when the host responds to these invaders, with white blood cells moving to the site and an inflammatory response. This means fever and other symptoms. Your doctor may need to prescribe an antibiotic or antifungal. However, not all antibiotics work against all bacteria. Your doctor will need to know the type of microbe or microbes causing the infection. The method used for more than 150 years to detect the bacteria causing your infection is laboratory culture. What are the problems with laboratory culture? Only 1 to 2% of all known microbes can be grown in culture. If the bacteria or fungus causing your infection does not grow in the culture lab, your doctor will not know what antibiotic or antifungal to prescribe. Using the wrong antibiotic for your infection will leave you to continue to feel miserable or even die. In chronic infections, the bacteria and fungus will form a collaborative community or biofilm. This means the microbes will form a wall or matrix to protect themselves and are more difficult to eradicate. The biofilm can form on any host tissue or implant within your body. Bacteria has been around for billions of years and have adapted. They can go into a VBNC state, viable but not culturable. They simply go into hibernation and will not grow in the culture lab. They are hiding, resulting in you not getting the right antibiotic. What diagnostic tool can overcome these issues around culture and getting bacteria to grow? Just like each of us has our own unique DNA, bacteria and fungus also have their own unique DNA. We now can take a sample from the host, urine, blood, tissue, fluid, swabs, and within that sample we collect the microbes living at that site of the infection. Rather than trying to grow them in a petri dish, we can extract their DNA. Like a crime scene, we collect the DNA and then compare the DNA to a known library or database of the exact DNA sequence code. A match of the bacterial DNA in from the sample to the database tells us what type of bacteria is in the sample. The DNA is the most precise method for detecting a microbe. Now that we know the type of bacteria, we can target the correct antibiotic or antifungal. Right bug, right drug. Using DNA to identify microbes is called molecular diagnostics. This type of molecular test is called next-generation sequencing, NGS. Only one lab, MicroGenDX, offers this type of technology and has a reimbursement code. MicroGenDX has the lowest price for the test and the fastest turnaround time. Your in-network lab, such as LabCorp or Quest, does not offer NGS for infections. As with anything new in medicine, the insurance companies are slow to adapt to new diagnostic tools and new antibiotics. Ask your doctor about MicroGenDX.
Good morning, everyone. Thank you very much for being here. And I'm going to go over culture versus next generation sequencing and what are the differences. Diagnosis of periparesthetic joint infection, PJI, is currently a very challenging problem. As you know, there is no absolute test. Hence, clinicians who encounter a suspected PJI case have to use a combination of tests. all of which, besides their expense, can be invasive, and even they are far from perfect. In 2013, the MSIs came up with a set of criteria for defining PJI, which was then modified by the International Consensus Meeting. And that definition also got modified in 2018 and now has a scoring system that includes a preoperative and intraoperative phase. However, all these criteria are heavily based on culture, because culture is part of the major and also part of the minor diagnostic criteria. And as Emanuele mentioned, culture negative PJIs are on the rise, and the literature shows that up to 34% of PJI cases, that's more than 1 out of 3 patients, are having negative cultures. So it's a huge issue for us that can compromise diagnosis and timely management of these patients. Culture is the traditional method of detecting infection and has been around for more than 200 years. And it's interesting because I cannot think of any other technology that has been around for this long without a major change. There are several disadvantages that are inherent to culture. One is the yield of culture is as low as 30%. And in order to battle this problem, we oftentimes obtain multiple samples to be able to increase the yield. Cultures are timely and they are costly because, again, we're taking multiple samples, and to run for a single patient, that just adds to the cost. And one of the other problems that oftentimes gets missed is that cultures take physical space. And I know at Cooper, our microbiology lab always complains about all the samples that we send, and we want to keep them for a long time for slow-growing organisms. And that's a challenge and a burden that is put on the microbiology labs. The other problem with cultures is that there is a lot of lab variation, and they are technique dependent, and different culture mediums can yield different results. And oftentimes, the results of culture can also be confusing because, let's say you send 4 samples for one patient and only one of them comes back positive on a random medium like thiobroth. It is extremely difficult to make a conclusion out of a situation like this because now you have 1 out of 4 that is growing something, so you cannot ignore it. At the same time, you have 3 negative results. So now whether this is contamination, is it false, like all those negatives are false negatives, these are all imposed challenges on a diagnosis that is already a challenging process. In order to unify the way the cultures are made and the cultures are processed and kind of synchronizing all the labs, the Infectious Disease Society of America and American Society for Microbiology came up with a set of guidelines. The main idea is that for cultures, you need to have a live microorganism so that it can grow. And looking at these guidelines, you can quickly see that we are not really following them because, for example, aerobic and anaerobic samples need to be transferred into a room temperature container immediately. And I'm sure that you have oftentimes seen the picture that I have in the bottom left, that the cultures are sitting in these boxes outside the clinic for almost 24 hours in 100 degrees in a summer day. So that just essentially kills the bacteria, so you cannot have a live organism to grow. There are several limitations that are mostly specific to orthopedics and to periparesthetic joint infection. One is the issue of biofilm formation. Again, for cultures, you need to have a live organism to be able to grow something out of it. And with synovial fluid aspirations, when biofilm is formed, the yield for capturing a live organism is extremely low. The other thing is that the time to diagnosis is extremely important for management of these patients. Oftentimes, you need to wait 14 to 21 days for cultures to conclude. And if you want to do a DARE procedure, you're going to lose that golden window just waiting for your culture results to come back. So now what's the solution? I think next generation sequencing can significantly help us with that. And in a nutshell, with next generation sequencing, we're matching the DNA and RNA molecules that are captured from a specimen and match them with a database that we have with the mapping of all the genomic information of these organisms. Just a little bit of story and history of NGS. It all started with the Human Genome Project in 1985. That was a study that was sponsored by NIH, Department of Energy, and Department of Defense with $3 billion at the time. That was the highest sponsored study of all times. And they tried to look at the genome and the map of the human genome. And almost 20 countries contributed to the study and collaborated on it. It took them about 10 years to have the complete mapping of human genome. And at a time, the cost was around $2 million, and it would take them 1 year to process and have the complete mapping. So as you can tell, it was not really a practical solution. But I want to grab your attention to the graph that I have on the right side of the slide. As you can see, in early 2000s, there was a huge drop in the cost, and that's where the next generation sequencing was introduced. And that's why now we have companies like 23andMe or Ancestry that kind of provide this test at costs below $100 just for the fun of it to see what your genomic mapping is. And I want to grab your attention to this slide, and that's essentially what next generation sequencing does for detecting infection. On the left is what culture is. You get a seed, and you want to make sure that that seed is alive. You plant it, wait for it to grow, and based on how it grows and the pattern of the leaves, you can tell me that it's maple. Or the alternative is what we have on the right side, have an atlas of all the pictures of all the seeds, and as soon as you pick it up, regardless of whether or not it's alive, you can match it with a picture of another seed that is in your genomic database and tell me right off the bat that it is maple, for example. And I think that's how NGS can significantly reduce the time and accuracy for diagnosis.
Thank you, Alexina.
My UTI was chronic.
I had a series of infections and problems.
All of the culture tests I took for years did not find the cause of infection.
I was a week and a half from them amputating my foot.
Across America, people needlessly suffer from chronic infections. Current testing methods typically use cultures, a method that presents limitations in identifying key organisms known to cause chronic infections.
Traditional culture has so many limitations.
Cultures have shortcomings.
Every day as I would see my patients, I knew I wasn't helping them because we were limited by traditional cultures.
MicroGenDX is a better test than culture because it uses the DNA from your urine, sinus, wound, or other infected sample and matches it to their database of more than 50,000 species of bacteria and fungi with a 99% accuracy.
MicroGenDX is not my gold standard, it's my only standard.
This has allowed me to do the right thing for my patients, give them the focused antibiotic therapy, and the results are better.
The regular lab came back negative, like it always did, and the MicroGenDX came back with exactly the bacteria that was there and what to treat it with.
I went to the doctor, and he recommended the MicroGenDX. So I was a week and a half away from not having a leg to being healed. It changed my life.
MicroGenDX gave me my life back. I was bedridden. I wasn't able to get out. I wasn't able to leave the house. Now I can be outside with the kiddos when before I couldn't. Thank you for giving me my life back.
Ask your doctor about MicroGenDX.
If you're suffering from chronic or recurring urinary, sinus, wound, or implant infections, ask your doctor to use the most accurate testing available with MicroGenDX DNA analysis. Go to microgendx.com for information to give to your doctor to help cure your infection and possibly save your life.
Thank you for tuning in to MicroGenDX Mic'd Up. We hope that you found our broadcast informative and helpful. If you have any questions or would like to learn more about our services, please visit our website at microgendx.com. Power up your precision with MicroGenDX testing, the key to accurate diagnostics and personalized treatments. Until next time, this is MicroGenDX Mic'd Up signing off.